Hi everyone, we're going to get started in about 15 seconds. We're just giving some others a chance to sign on. Welcome to Metastatic Breast Cancer and the Tumor Microenvironment. I'm Christine Benjamin, and I'm the Breast Cancer Program Director at SHARE. Before the presentation begins, I'd like to tell you just a little bit about SHARE. SHARE is a nonprofit organization that has been helping people through breast and ovarian cancer for the last 43 years by offering the support of those who have been there. SHARE offers many services, including a helpline, telephone and in-person support groups, and educational programs. Our services are free of charge to participants. For more information, please visit our website at sharecancersupport.org. All participants will be muted during the presentation. When Dr. Ghajar finishes speaking, we'll begin the Q&A discussion. You're welcome to submit questions during the presentation through the question pane in the control panel on your screen. This webinar is being recorded and will be available on the SHARE website soon. So now I'd like to introduce our speaker. Dr. Cyrus Ghajar is an associate member of the Public Health Science Translational Research Program at the Fred Hutchinson Cancer Research Center. Dr. Ghajar researches metastasis and how the microenvironments of these sites, including bones, can put cancer cells to sleep or wake them up. Dr. Ghajar directs the Laboratory for the Study of Metastatic Microenvironments. The goal of his research program is to understand how microenvironments within distant tissues regulate dormancy, growth, and drug resistance of disseminated tumor cells. His belief is that solving these puzzles will allow the development of therapeutic regimens that eradicate dormant disseminated tumor cells before they can develop into full-blown metastasis. Please help me welcome Dr. Ghajar.
Hi, everybody. Thank you. Thanks for the introduction. So my name is Cyrus Ghajar. I'm going to talk to you about metastatic breast cancer and the tumor microenvironment. And I've tried to structure this so that I give extensive background and then talk about my laboratory's more recent research about how we can target tumor dormancy. And I think that hopefully the first part of the talk will really illustrate why we want to target tumor dormancy, and then the very last part we'll talk about how we're going about that. I'd like to stay out of the weeds and more kind of into the overview, so I'm going to try my best to do that. And I tried to structure this so that we're done around a quarter until the hour, so that leaves about 10 to 15 minutes for questions. So, all right. So, how did we come to appreciate the microenvironment? I think one of the earliest, not the earliest, but one of the earliest examples of why we really needed to pay attention to the microenvironment is a very famous hypothesis put forth by a British pathologist named Stephen Paget. And he published this article in the Lancet journal back in 1889, so 130 years ago, and put forth this hypothesis that is now known as the seed and soil hypothesis. And what he did was look at 735 fatal cases of breast cancer, where these women succumbed to metastatic disease, and then said, with regards to metastasis, here too we shall find evidences of predisposition. We shall see that one remote organ is more prone to be the seat of secondary growth than another. And he specifically pointed out that he time and time again would see an enrichment of metastases to the lung, to the liver, and to the bones, and also to the ovary. So what was actually quite notable was back then, 130 years ago, central nervous system or brain metastases were actually quite rare. This was actually substantiated by an autopsy study that was performed about 20 to 50 years later. And I think that that's really been, brain metastases have become a lot more common now because we have better therapies that have allowed patients to live longer. And now as people are living longer, brain metastases have emerged more and more. But the main point here was that there is a tendency of breast cancer cells to not only spread, but to spread and grow at specific organs. And this really brought about the notion of paying attention to why is it that liver, lung, bone are such favorable environments for breast cancer cells eventually to emerge. So what is it that led us to really believe that the microenvironment could be important? If you're going to say that the microenvironment, or the cells, the normal cells and tissues around a tumor cell, are extremely important, you want to have two different examples. One is that you want to be able to show that the microenvironment can actually regulate whether or not a tumor cell becomes a tumor. And you also want to show that the microenvironment can actually force a normal cell to become a tumor. So this is an example of the first. And this is a virus that is the first discovered oncolytic or oncovirus. This was discovered by Peyton Rouse over 100 years ago. And this virus called Rouse sarcoma virus encodes the first discovered oncogene called pp60-sarc. And the virus would go in and infect cells. And when you injected, how they showed this was that you would take the sarcoma cells, spin them down, and then you get a pellet at the bottom of the tube. If you took everything other than the cells, so everything floating above the cells, and you could inject an animal with that, what's called the supernatant, you could actually cause tumors in that animal. So that told you that there was something floating around that the cells were secreting, and that's how they ultimately isolated a virus. So Mina Bissell in the early 1980s took this virus, labeled it with something that allows you to make it blue. So you can hopefully see my arrowhead, and you can see all these blue nuclei. And then they injected chickens, and they showed that if you inject the embryo, you have a number of cells that take up the virus and actually are expressing this oncogene. But you can see how beautiful this feather looks, that they would not transform and form a tumor. So what that was telling you is that there was something special about the embryo that was actually able to suppress these tumor cells and not allow them to form a tumor. So there's something special about the embryonic microenvironment that suppresses tumor progression. What they also showed that was really quite fascinating was, now if you took adult chickens and you injected them, they noted that the chickens only develop tumors at the site of injection. So they would inject them in, let's say, the right wing, and then they would only get tumors in their right wing. And they said, well, that's really strange. And having this blue label allowed them to see that even the cells in the left wing were taking up the virus, yet you would not see tumors in the left wing. So they said, what's different between the right wing and the left wing? Well, we're injecting the right wing. Maybe there's something about the injection itself, sticking a needle into the right wing, that caused tumors to form. So to test that, they took a clip and then they actually cut the left wing, the non-injected wing of the animal, and they showed here this really gruesome tumor would form, and that was the result of wounding. So despite the fact that you have cells that are expressing an oncogene, they're only able to progress with a second hit, in this case to their microenvironment, and that is causing a wound. So this is already showing you that the microenvironment is really quite dominant. A suppressive microenvironment can prevent cells with an oncogene from progressing, and you need a permissive microenvironment, in this case wounding, to allow a tumor to form. Can a microenvironment on its own take normal cells and actually make them tumorigenic? This was a paper that was done again by Mina Bissell and Zena Werb and at UCSF. And then what they show here is they've taken an enzyme called MMP3 and expressed it only in the stroma, not the epithelium. This is the mammary epithelium, which will ultimately form tumors. They're not expressing an oncogene in mammary epithelium. They're expressing an enzyme only outside of the mammary epithelium. When that happens, you can focus on these black bars here, expressing transgenics. You can see how much fewer animals have normal mammary glands, how much more fibrosis you have, how much more infiltrate you have, how much more hyperplasia and dysplasia, the initial phases of tumor formation or of disorganized tissue you have in these mice expressing an enzyme in their stroma, and you actually see an incidence of carcinoma or mammary cancer in these mice. Again, not by making mammary epithelial cells express an oncogene or a deleted tumor suppressor, but by expressing an enzyme that disorganizes the microenvironment in the stroma of these animals. So what can we conclude from these really illustrative studies? The microenvironment can suppress tumorigenesis, An oncogene is not enough. The microenvironment can also promote tumorigenesis, even in the absence of an oncogene, at least initially. MMP3, this enzyme, eventually causes chromosomal instability, and you need an oncogene to actually get mutated and then activated in these cells to get a tumor, but initially you don't need that. You can have a messed up microenvironment that can actually cause the cancer. So context is everything, and this applies to metastasis also. So before we proceed, I really want to define what we consider a tumor microenvironment, and this is the other normal cells like fibroblasts, like endothelial cells, like macrophages and other immune cells. Insoluble factors like extracellular matrix, like collagen, for instance, soluble species, which include hormones that surround a tumor cell. And this can even be other epithelial cells, and really for the tumor microenvironment, other tumor cells can be part of a given cell's microenvironment. I also would just say, though we're not going to talk about this a lot today, that you also have to consider the systemic nature of the disease, meaning that even though we're talking about breast cancer, what happens in the liver is important, what happens in the brain is important, what happens throughout the body is important, and also the macroenvironment is important. The interactions that we have in our environment, whether or not we feel fulfilled, whether or not we're actually having fun. I mean, this sounds kind of crazy, but they've housed mice that are prone to get cancer on their own or with other mice, or with toys and without toys. And when you do these things, when mice are with other mice, they get less tumors and it takes longer. When mice are alone, they get more tumors faster. When mice have toys, they get less tumors and it takes longer for them to get those tumors. Without toys, the opposite. So these are all kind of important things. So how does the microenvironment influence metastasis? That's going to be really the focus today. Metastasis is Greek for displacement. So you have a tumor moving from one area of the body to another. And this refers to the spread and growth of a tumor. So if the tumor is only spreading but not growing, that is called dissemination. When the tumor cell is leaving the breast, going to the liver, and then ultimately growing or colonizing the liver, then that actually is called metastasis. So this is how the metastatic cascade is typically depicted. You have a cancer that originally could be in situ, meaning it's contained within a tube. It becomes invasive and breaks through a layer of that tube called the basement membrane and into the surrounding stroma. It needs to recruit blood vessels in order to continue growing. It invades blood vessels, circulates throughout those blood vessels, and ultimately leaves those blood vessels. And then if they survive and ultimately can grow at that secondary site, they colonize that site. And that is the final step of the metastatic cascade. So what we're going to do for each of these steps, invasion, angiogenesis, intravasation, extravasation, or entry and exit from vessels, and colonization, is talk about what cells need to be able to do this, how they actually get help from their friends, i.e. the microenvironment. And despite the fact that we like to think so dogmatically and say, well, this is what has to happen, this is what has to happen, and this is what has to happen, I think in a very non-dogmatic way. I don't believe in dogma, I believe in breaking rules, and I'm going to give you an exception to each of the quote-unquote rules in cancer biology. And so that should really hopefully encourage a lot of you to question what you hear and really push people when they're telling you something has to happen all the time. And you can ask them, does it really have to happen all the time? Are there any exceptions to this, and what can we learn from those exceptions? So first we're going to start with invasion. This is a frozen cast of a tissue, and this kind of beige object here is a couple of tumor cells, a few tumor cells invading their surrounding matrix. And you can see how much of a barrier these cells actually need to go through in order to penetrate the tissue. And it's really like trying to cut your way through a jungle. So if each of you imagine that you were, I dropped you into a really dense jungle like this blue area here, what you would need to be able to move and get yourself out of it. You might think that you need something to attach to, you need to grab something, you need something to pull yourself through this environment. And when it gets too dense, you really need something to cut through the jungle. And cells have all of these things. They attach through molecules called integrins, which are going to be kind of a recurring theme of this presentation. So integrins are receptors that integrate extracellular cues into intracellular biochemical signals. So they attach to the matrix around a cell. They have contractile machinery, basically a skeleton inside the cell that allows it to pull itself through the environment. And then they have machetes to cut their way through the jungle. And those, one example of these machetes are enzymes called matrix metalloproteinases, or MMPs. And we were talking about MMP3, which is one of these MMPs a little bit ago. So integrins. Integrins are receptors that integrate extracellular cues coming from the extracellular matrix into biochemical signals inside of the cell. They're dimers, meaning they have two portions to the molecule, and they're heterodimers, meaning that these two portions are actually different. There's an alpha subunit and a beta subunit. And what these alpha and beta subunits do is actually tell the cell what it's binding to. It's kind of like the tissue is a lock, and then the alpha beta combination is a key to that lock, or a combination to that lock, where specific alpha subunits dictate, for instance, that the cell with beta 1 integrin is going to bind to collagen, whereas different alpha subunits are going to bind to another type of extracellular matrix molecule called laminin, and you have different alpha beta combinations that bind to other things that are in extracellular matrix. And this is really complicated, and we're not going to get into it, but the point is that when a cell binds the extracellular matrix, this binding actually triggers a whole cascade of signaling that can tell a cell whether it should survive, whether it should divide, or whether it should move. And this is very similar to growth factor signaling. So many of you may be familiar with epidermal growth factor or inhibitors of epidermal growth factor, EGF receptor, like lapatinib, or something like HER2 or HER2 inhibitors like Herceptin, and how they're targeting growth factor signaling. Well, integrin signaling is very similar and overlaps a lot with growth factor signaling. So by binding the matrix around you, you can actually get a very similar interpretation of that on the level of the cell as binding one of the ligands for a growth factor receptor. Once the cell is bound, as I said, it amplifies a signal. So how does it do that? It builds something called a focal adhesion. So here's the integrin in red, and it's binding this cyan extracellular matrix. And one integrin then has a bunch of molecules that attach to it, which then have a bunch more molecules that attach to that, which then have a bunch more molecules that attach to that. So what it's doing is taking a localized signal of binding a piece of tissue and then transducing or amplifying that signal into this biochemical cascade that then tells the cell that it can survive. If it gets enough of that signal, it may grow. If it gets a different kind of signal, it may actually start moving and invading. So that starts to tell you how these tissue cues can actually turn into changing cell behavior. Here's a really nice example. So right underneath these integrins is the focal adhesion layer, and there's this molecule called FAK, which is called focal adhesion kinase. Here is a fibroblast that's attached to plastic and proteins on the plastic. You can see how nice and spread they are. You just take away FAK, and then look what happens. They can't spread at all. They're attached, but just can't generate any tension and activate their skeleton to basically spread. And this is important. If you can look at, this is a breast tumor here in the top left corner. Tumor that can't organize its collagen around itself and actually pull it in like this one. This collagen that isn't getting pulled in actually functions like a belt and prevents the tumor from invading. When the tumor starts yanking on the collagen and it organizes it perpendicular to its surface, then it makes it easier for these cells to invade. Breast tumors from patients that have this signature are less likely to be metastatic. Breast tumors that have this signature somewhere in the patient's tumor are actually more likely to be invasive and metastatic. And that's work by Paolo Provenzano and Patricia Keeley, who have done that work. So now you can attach to the matrix. You need to be able to yank on the matrix, and that can allow you to pull yourself through, just like you want to pull yourself through this jungle, right? But ultimately, that jungle gets too dense. You can't move through it until you actually start cutting yourself a path. And the way that these cells do this are through enzymes called matrix metalloproteinases, or MMPs. There are a lot of different kinds of MMPs, about 26 different ones. Remarkably, it's thought that only one of them is really critical for a cell to be able to cut its way through the matrix, and this one is called MT1 MMP. So you can see breast tumor cells in a collagen matrix at day 0 here in red, and then 3 days later, you can see how invasive they are. If you take away some MMPs, they're still really invasive. If you take away MT1 MMP, they are not able to migrate through that collagen anymore. So these things are, they really, when that jungle is thick enough, they really need these machetes to be able to cut their way through. And if they don't have that, then they're not going to go anywhere. So how does a tumor cell still invade, perhaps in the absence of MMPs? Those of you who are really familiar with how breast cancer treatment's gone know that there was a lot of hype about MMP inhibitors, right? This looks amazing. You would say, oh, all we need is to inhibit this MMP. Breast tumors will never invade. Everything is going to be okay from that point forward. But that really didn't go anywhere. The drugs were toxic, and they also were not nearly as effective enough. And the reason is because despite taking this as dogma, again, like a lot of people would want you to, the fact is that tumors can actually get through matrix through other ways. And one way they do that is by actually getting help from cells in their microenvironment. So this is a really neat example I'll try to walk you through. This is a skin carcinoma, okay? So this is like a layer of skin, and this is modeled actually in culture, so in a dish in the lab. And you can see that when you combine your skin tumor cells with matrix underneath that has fibroblasts in it, these cells are able to invade. If you separate your skin tumor from your layer of fibroblasts with another thin layer of matrix, look how nice and thin these tumor cells are. They look totally like normal skin, and they're not able to invade. The key is that these fibroblasts are actually making tunnels for the tumor cells to invade, so that they could actually invade in the absence of MMPs. This is a gel that was just treated with detergent and then seeded with these tumor cells. Nothing happens. This is a gel that had fibroblasts in it. All the fibroblasts were killed with the detergent, and then you seeded the tumor cells on top, and now you can see that they're able to invade again without the fibroblasts actually being there. Those fibroblasts dug tunnels. Even once they're dead, these tumor cells are now able to invade through the tunnels. And so what happens is the fibroblasts labeled in red can actually grab onto the tumor cells in green and pull them through these tunnels. So you can imagine that if this video were playing, you could actually see these fibroblasts pull the tumor cells through the tunnels. So in these cases, maybe you actually don't need the machetes to get through the jungle when you have this other cell that's able to pull you through the jungle itself. So an exception to the rule, MMPs may not always be necessary. This is a paper from Peter Friedel's lab. And what they're showing is that you can inhibit all kinds of proteinases, basically take away the machete from a cell, and the cells can still wiggle their way through the matrix. And they're literally wiggling their way through. So you can imagine the bigger the tunnel, the easier they're able to wiggle. And the rate limiting step, or the limiting step to be able to wiggle through the matrix, is how deformable the cell's nucleus is. The more it's able to deform its nucleus, the better it can wiggle through. The less it can deform its nucleus, the less it can wiggle through. And so really these cells can get through a dense jungle as long as they're able to kind of contort themselves in a way to be able to do that. So that tells us a lot about invasion, and now we've gotten through, and the tumor needs to grow around into the surrounding tissue. And ultimately we're taught that the tumor needs to feed itself. In order to feed itself, it needs to give itself blood vessels, and that's a process that of forming new blood vessels from pre-existing vessels called angiogenesis. So again, those of you kind of familiar with cancer treatment will be really familiar with how targeting angiogenesis was a very, very hot thing to do in the tumor field as recently as 10 years ago. So angiogenesis is the formation of new microvasculature from pre-existing vasculature, and why we began to think it was so important was that scientists at Harvard named Judah Folkman did experiments where he took a tumor cluster and put it on the eye of a rabbit. When he put it far away from blood vessels, the tumors wouldn't grow. If you put it close enough to blood vessels, the tumors would recruit blood vessels, feed themselves, and ultimately grow. So he put forth this hypothesis that if we can hold a tumor away from vasculature, it'll stay in a dormant state. And you could think then if you're able to target the vasculature, prevent it from growing, that you could actually keep tumors dormant forever, and that this is something that we should really explore. So he said this back in 1971, which is really quite prophetic in many ways. So there was evidence in a number of different tumors that there are vascular hotspots or regions of the tumor that are really dense with vasculature. And then there were a number of molecules that were discovered to actually regulate what's called the angiogenic switch, where you have a lot of pro-angiogenic molecules. You tilt the teeter-totter in this direction, and then you get blood vessel formation, and you need less of the inhibitors of angiogenesis or the inhibitors of blood vessel formation. And again, that helps you get the switch going in the right direction for the tumor to then become vascularized and ultimately grow. So why hasn't this worked? There are a lot of other cells in the microenvironment of tumors other than the tumor cells that secrete pro-angiogenic molecules. So I circled pro-angiogenic here, and this is just from a review showing that macrophages, so immune cells, monocytes, which are macrophage precursors, neutrophils, another immune cell type, mast cells, which are like the very first cells that come into a wound anytime you cut your skin, platelets, which are all throughout our blood, fibroblasts, all of them also secrete molecules that help promote blood vessel formation. You can imagine that when you cut yourself, this is a very important thing that needs to happen, is that you have to repair the blood vessels and ultimately feed the new tissue that is going to form. So there's a lot of other ways that tumors can become vascularized that don't really depend on inhibiting what the tumor cell is secreting. And there's a diversity of molecules that allow this to happen. So just inhibiting one may not be enough. Another thing that happens, which is really quite remarkable, is that tumor cells are plastic enough to actually form their own blood vessels. So tumor cells can actually wrap into tubes and become vessels that then feed the tumors. This is a process called vasculogenic mimicry, which was first described by Andy Maniotis and Mary Hendricks back in 1999. And so you can see that these tumor cells here that aren't staining positive for a lot of endothelial markers are actually forming networks that then help feed the tumor. And there's some thought that tumors that exhibit this behavior are actually a lot more aggressive and portend to poor survival than those that aren't so, plastic or able to change their behaviors in such drastic ways. So we've talked about invasion and angiogenesis. After the tumor's formed and is able to grow, they need to get into vessels and they need to get out of vessels. So we're gonna go over these things very quickly together because it's this complicated cascade of getting in and out of vessels. And the bottom line, really what you need to know is that tumor cells can go through the cells that make up blood vessels called endothelial cells. They can go right in between them, or they can actually cause these cells to form kind of donuts and go right through the middle of them. And when they get out, they really behave like immune cells. They roll along the vessels and they ultimately get out in a very similar way as they get into the vessels. And that requires a whole lot of molecules, and it also can involve some help from the tumor and cells in the microenvironment. So one way that the microenvironment gets disrupted by tumors is that the cells that cover blood vessels called pericytes, which are here in red, actually get stripped off of vessels. So you can see all these examples of green vessels that are not covered with red as they otherwise should be. And so that allows the tumor to have very easy access to the blood vessel where otherwise it wouldn't. And once they're trying to get out of the vessel, they can actually get help by cells like macrophages. Specifically, there's one type of macrophage called an inflammatory macrophage that can actually help pull the tumor cell out of the blood vessel. And so there's some evidence that if you block this kind of macrophage with an antibody, that you can actually reduce the number of tumor cells going through the vessels and coming out into a tissue like the lung, for instance. So what's an exception to being able to get in and out of blood vessels? I mean, there maybe aren't many in breast cancer, although inflammatory breast cancer may be kind of one type of exception because the breast tumor cells actually can grow along vessels rather than going in. Brain tumors actually really do this as well. So brain tumors don't metastasize outside of the brain. There's not certain whether they or not they kind of like go into the vessels and come out inside of the brain. But one thing we know is that they're actually able to migrate along blood vessels into the brain and then go from one part of the brain to another. That's why when you cut out a brain tumor or even a brain metastasis, that doesn't help survival so greatly because there are all these finger-like projections throughout the brain. Once you've removed one tumor, then those finger-like projections can give rise to another tumor deeper into the brain, somewhere where we really can't operate. And so that's a really big problem. So we've gone through invasion, angiogenesis, intravasation, extravasation. You might wonder, well, we know a lot about how a tumor cell accomplishes all of these steps. And as you can imagine, having all this knowledge has led to a lot of drugs that try to target and block invasion. They try to target and block angiogenesis. We've even tried to target cells getting in and out of vessels. And ultimately, that hasn't really worked. And so you might ask yourself, why? And the reason is because when tumor cells, or when a tumor is already detected in a patient, when a breast tumor is detected in a patient, there's evidence that all of these things have already occurred. So blocking invasion, angiogenesis, intravasation, extravasation, it may be too late. And there's even evidence that tumor cells may have actually exited so early or left the tumor so early that even earlier detection may not help. One of the best examples of this is this really, really important study that was published over 10 years ago now by Christoph Klein's lab in Germany. And what they showed in a mouse that gets tumors around 22 to 26 weeks of age, so about 6-month-old mice get these HER2 positive mammary tumors. If you go as early as week 4, where there's no tumor at all, and the oncogene that the mice need to get a tumor is only just activated, you can already find tumor cells in the bone marrow of these mice. So these are cytokeratin positive, HER2 positive tumor cells in the bone marrow of mice as early as 4 weeks of age. And despite the fact that this tumor grows and now you have orders and orders and orders of magnitude more tumor cells in the mammary gland, you can see that the number of cells in the bone marrow is really not increasing drastically. So that tells you that not only are these cells able to get out early, but they're also able to establish themselves in the bone marrow with remarkable efficiency. So so you have to go back to the mammary gland and use electron microscopy to even see any signs of invasion at 4 weeks of age. And so the mammary gland, the point is the mammary gland of this mouse is remarkably normal at 4 weeks of age, yet these initial cancer cells are still getting out and then establishing themselves in the bone marrow with remarkable efficiency. So does this happen in humans? This is one of these cases where work in a mouse really prompted us to look more closely in humans. We know from this really large scale study that was published in the New England Journal of Medicine by Klaus Pantel's group that in stage 1 patients, 25% of those with localized tumors exhibit detectable cells in their bone marrow. But then a more recent study of patients with DCIS or ductal carcinoma in situ, so supposedly non-invasive breast cancer, anywhere from 20 to 50% of these patients, this is a much smaller scale study, but anywhere from 1/5 to 1/2 of these patients had cells in their bone marrow, despite having supposedly localized disease. Now, what's important to realize is that many of these patients are not going to progress. So the majority of these patients with cells in their bone marrow will not progress, but those with cells in their bone marrow, those patients have a much higher risk factor still than patients who don't have cells in their bone marrow of eventually recurring. So that's probably something we should talk about after we go through the rest of these slides. So now we know that cells are leaving early, and we also know that they're showing up later and later. So this is a study of patients who took endocrine therapy for 5 years and then were followed for an additional 10 to 15 years. And you can see now this yellow line is patients who don't have any involvement in their lymph nodes. Blue is 1 to 3 lymph nodes involved, and red is 4 to 9 lymph nodes involved. At year 5, you have 6, 10, and 22% of patients who had distant recurrence. At 20 years later, it's estimated, or 22% of patients with no lymph node involvement now have recurred. So the vast majority of patients are going to recur 5 years or more after the initial diagnosis and treatment. It's the same kind of thing if you have more lymph node involvement or a lot of lymph node involvement, where these endocrine therapies aren't curing breast cancer necessarily, but they're pushing off recurrence to a later date. And what that tells you is that we need therapies that are actually targeting the root of metastasis, the cells that have left early, established themselves in the bone marrow, in the liver, in the lung, in the brain, and then ultimately are somehow waking up later and then causing metastases. We need to target those cells before they actually sprout and then and cause metastases. Right now, there's not a single therapy that targets these cells, and so that's really the focus of my laboratory. How can we target cells that have left the breast before they actually do anything to kill a patient? How can we either keep them dormant forever, or how can we kill them before they ever have a chance to grow and form lethal metastases in places like the brain, lung, liver, or bone? So what we know is that when cells have left the breast and before they become these lethal metastases, they can exist in distant organs as masses, like what Judah Folkman was showing, of cells that die and grow at the same rate. So you can say that they're steady state, or they're a stable population, and they're waiting on blood vessels or immune invasion in order to progress from this state to this state of a full-blown metastases. But Ann Chambers also showed that tumor cells can actually exist as single cells or very small clusters of cells in these distant organs. And the cells have exited the cell cycle, they're not dividing, and they're in a state of dormancy called quiescence, which means the cell has exited the cell cycle and is not dividing. And so which is more important? Is it these masses that are growing and dying at the same rate, or is it these single cells that have just fallen asleep and are sitting there? The truth is we don't know right now, and it very well may differ based on if you're looking in a bone marrow, or if you're looking in a brain, or you're looking in a lung, or you're looking in a liver. But what we do have access to as a scientist is bone marrow, because you can stick a needle in someone's hip and you can suck out bone marrow, and then you can stain for markers of breast tumor cells. And when you do that, by and large, what you find are single or very small clusters of like 2 to 4 breast tumor cells. These express cytokeratin, which other normal cells in the bone marrow don't. And so you're able to say, okay, here's a breast tumor cell, and there's one tumor cell. So by and large, this is what we find. And if you take an aspirate 3 years down the line, 5 years down the line, this is likely what you would find, even though that patient might get metastases 15 years down the road. So that suggests that these metastases are ultimately coming from single cells, and which tells you that maybe we want to target these single cells and kill them before they ever have a chance to form metastases. So how can we do that? Well, the cells leave the breast really early on, and then they can survive in a tissue like the bone marrow for 10, 15, 20 years. So we could try to figure out how they survive and then deprive them of those signals and then kill them and then prevent them from forming metastases. That's one thing that we're working on, and I'm not going to show you those data today. We could try to understand how they're put to sleep, and then we could do things to actually reinforce those signals and keep the cells asleep for good and prevent them from waking up. And so that would be one really great approach. The downside being that you have to live with these cells in your body, but if we really were confident that they were put to sleep and that they were going to stay asleep, that would be fine. The other thing that the other property that they have that we can target is the fact that they're resistant to chemotherapy. A lot of people overlook this, and they assume that the cells are resistant to chemotherapy because we're told time and time again that chemotherapy only targets rapidly dividing cells. So we really wondered, is that the case, or do these dormant cells escape from chemotherapy because of something in their microenvironment? And this was important to us because you could imagine if dormant tumor cells are resistant to chemotherapy because of something in their microenvironment, then you could actually target them with an adjuvant or a therapy that's given around the time of surgery and afterward in conjunction with chemotherapy in order to make the disseminated tumor cells in their bone marrow and elsewhere sensitive to chemotherapy. You would eliminate the reservoir of disease and then prevent these metastases from cropping up 5, 10, 15 years later. And so what we wanted to do is to see whether or not there really was something in their microenvironment or their niche that was protecting dormant disseminated tumor cells from chemotherapy. So what we did is basically take mice and treat them just like patients with invasive breast cancer. They had a breast tumor that was surgically removed, and then they were given dose-dense chemotherapy, doxorubicin and cyclophosphamide for 5 cycles weekly, and then we looked at these mice a week later to see what we could find. And so we looked in their bone marrow, and this is a single dormant tumor cell here. And for each one of these cells, we measured their distance to blood vessels, their distance to these other cells in the bone marrow called megakaryocytes, which are the cells that make a bunch of platelets. And we also measure their distance to the osteoblasts, or the bone-forming cells of the bone. So you'd say, why did we measure their distance to these cell types? These are the 3 cell types that have been implicated in dormancy of our blood stem cells, which live in bone marrow and occupy very similar environments as these breast tumor cells. So what we found is, so this is 350 tumor cells. Each one has 3 dots. A red dot for its distance to the blood vessel, a gray dot for its distance to the megakaryocyte or the platelet producing cell, and black dot for its distance to the osteoblast or the bone forming cell. And you can see right away that all of those red dots collapse down onto the x-axis, meaning these cells are very, very close to blood vessels. And these are 3 images of 3 different cells, and you can see how close they are to blood vessels in the bone marrow. So what this was telling us is that maybe, because chemotherapy was killing some of these dormant cells, and so we had less cells in chemotherapy-treated mice than untreated mice, and they were localizing to the vessels after treatment. So maybe the blood vessels are actually protecting dormant cells from chemotherapy. So how could we actually show that? Well, what we did was build a bone marrow microenvironment in a dish, and we did that by taking bone marrow stromal cells and endothelial cells, or the cells that make blood vessels. And we actually made vascular networks in a dish. And you can see now when we see these networks of bone marrow vasculature with tumor cells, the tumor cells are going to be in white. Okay, so these are the vessels forming actually like tubes. So you can see they're actually hollow inside and like they could carry fluid inside of them. So now we seed breast tumor cells. If we only seed them on stroma in the absence of blood vessels, you can see how much they outgrow. If we seed them on stroma and blood vessels, you can see how much less they grow. So these endothelial cells are really important for actually driving these cells into a dormant state and keeping them there. So now what we did was seed tumor cells either on stroma or stroma and blood vessels. We let those tumors form over 12 days. They grow a lot on stroma. They don't grow very much on blood vessels. And then we treat with chemotherapy and see what happens. The blue is labeling DNA, and then the red is labeling broken DNA. So cells that are dying are going to have broken DNA, and they're going to have a lot of red in their blue nuclei. And so you can see that the amount of red is increasing with the dose of chemotherapy, and so we have a lot of death with chemotherapy when we don't have blood vessels present. When we do have blood vessels present, you can see there's no increase in red here. So cells are not responding to chemotherapy when they're around blood vessels. And so one kind of thing that's important is that the cells around blood vessels are also typically dormant. So chemotherapy targets rapidly dividing cells. We can actually make cells grow on blood vessels by adding a growth factor. And even when we do that, they don't respond to chemotherapy. So we're able to separate growth from growth from microenvironment. And even when we make cells grow around blood vessels, they don't respond to chemotherapy. So that's telling you that the blood vessel is doing something to protect cells from chemotherapy. What is it? This is kind of a complicated slide, but the bottom line is we did something called RNA sequencing, where we profile all of the RNA that eventually becomes proteins in these niches, the niches with blood vessels and the niches without blood vessels. And we asked, what is it around blood vessels that could be protecting these cells from chemotherapy? And this list is really what we found that was enriched or deposited or expressed more highly around blood vessels. And there was this family of genes involved in integrin binding that came up. These genes encode a number of proteins that get deposited on the outside of blood vessels. And so we thought maybe the dormant tumor cells are attaching to blood vessels or attaching to some of these proteins on blood vessels. And that signal is really important for these cells to survive chemotherapy. And we were talking about integrins before, and it turns out that integrins on the surface of these dormant tumor cells are needed to bind a lot of these proteins. So we said, okay, let's inhibit the proteins, let's inhibit integrins so that the dormant cells can't bind to any of these proteins. We inhibited a bunch of different integrins, and what we found is that, and so we wanted to inhibit the integrins prior to chemotherapy and during chemotherapy. And what we found is that when we inhibited 2 different integrins, one called integrin beta 1 and one called integrin alpha V beta 3, we went from these dormant cells that wouldn't respond to chemotherapy, no red in their nuclei, to dormant cells that would respond to chemotherapy. You can see there is red in their nuclei. And when we added the antibodies together, we got more death. So this is a single cell that is dormant. You see very little death, about 1/3 of them die with chemotherapy. When we inhibit one integrin, we get more death. We inhibit the other integrin, we get more death. When we inhibit them both together, we go from killing 1/3 of our cells with chemotherapy to killing over 90% of our cells with chemotherapy. That's single cells. Clusters, we get very similar data, and these drugs are not actually making these dormant cells divide. If anything, just adding the drugs on their own is making the dormant cells even more dormant. They're even less of the cells are dividing. So now the question is, this is all in this model I was showing you before that's in a dish. Going back to that, this is all in this model that's in a dish. So what happens if we actually go into a preclinical model and look at whether or not we can kill dormant cells and whether that's good enough to prevent bone metastasis? So that's what we did. So we did a bone metastasis prevention trial. We took overectomized mice. So we're now suppressing estrogen. We're putting in ER-positive breast cancer cells. And then we're treating with this integrin blocking antibody or combination of integrin blocking antibodies and chemotherapy, or just chemotherapy on its own. And then we're looking in one cohort at the dormant cells in their marrow, and in another cohort, we're imaging for metastasis-free survival. This is the bone marrow of a mouse that was just treated with chemotherapy. All of the green are the tumor cells. So you can see how many tumor cells are here at the end of the bone and how many tumor cells are here in the middle of the bone. So there's a lot of tumor cells still left after chemotherapy. This is the bone of a mouse that was treated with the integrin antibody and chemotherapy. And you can see what a great job it's done at eliminating all of the green. You just have a few green cells left over after chemotherapy. This is both of the integrin inhibitory antibodies and chemotherapy. It basically does the same thing. So combining integrin inhibition with chemotherapy depletes 94% of the cells in bone marrow. And now the question is, okay, that's great, but will it actually prevent metastasis? And the answer is that yes, it does. Just one integrin antibody on its own, the one that inhibits integrin beta 1, is sufficient to prevent bone metastasis. Chemotherapy alone, 70% of the mice recur. Chemotherapy and integrin inhibition, only 22% of the mice recur with bone metastases over a 15-week period. We chose 15 weeks because it's like the analog of about 10 years in a human based on the lifespan of a mouse and the lifespan of a person. So the remaining question is, does combining integrin inhibition with chemotherapy actually make chemotherapy worse in terms of toxicity? And granted, we're studying mice here, which are obviously very different from humans, but we didn't see any evidence of changes in body weight with chemotherapy on its own or combined with integrin inhibition. We didn't see any difference in the vascular density, and we didn't see any drastic differences in bone marrow cellularity, meaning immune cell phenotypes that are being skewed with chemotherapy are not being skewed anymore when we add integrin inhibition to the chemotherapy. So what we can say is that, at least in mice, there's no exacerbation of chemotherapy-associated toxicity by adding integrin inhibition to chemotherapy. So I'll wrap up there and leave us about 10 minutes for questions. So I want to just say really fast that transformation or an oncogene on its own is not sufficient to cause a tumor to form, but that progression actually requires a permissive microenvironment. So our tissues, when they're healthy, are actually very resistant to tumors progressing, but once they're compromised, they are more permissive to a tumor progressing. The microenvironment can be co-opted or actually used by the tumor at every step of the metastatic cascade: invasion, angiogenesis, intravasation, extravasation, and even colonization. But we can't target every step because dissemination is a very early event. So we really have to focus on targeting dormancy or these later events. The niche around vasculature is very critical to keeping disseminated breast tumor cells dormant. And we can target this niche to make dormant tumor cells sensitive to chemotherapy, which prevents metastasis without exacerbating chemotherapy-associated side effects. So we'll end there, and I'm happy to take any questions at this point with the time that we have left. So thank you all for listening, and let's open it up for discussion.
Thank you so much for that really incredible presentation and for letting us into the work that you're doing.
My pleasure.
Everyone who's listening, please submit questions through the question pane. Currently there are no questions, but we have some that came in earlier. You may or may not have answered some of these, but I think it's good review because there was a lot of information. Has your research program made breakthroughs that might help translate this area of work into potential therapeutic approaches?
Yeah, so I think that the key finding that I showed here is this fact that dormant cells, is common assumption that dormant cells are not responsive to chemotherapy because they're not dividing, is false. So even if we hadn't identified any molecules that are making the cells resistant to chemotherapy, that's a concept that I hope is going to spur others to really start investigating how the cells are actually resistant to chemotherapy and how we can make them sensitive to chemotherapy. And now the identification of this interaction between the dormant cell and the blood vessel that's mediated by this protein called integrin beta 1, I think is really important because it then allows us to now start pursuing an approach where we try to humanize an antibody to integrin beta 1, which means that we're going to take an antibody and make it human-like so that it can actually be put inside of a person. And if that in and of itself is not toxic, then we can think about combining that with chemotherapy for somebody who's going to already get chemotherapy and see if that's actually capable of eliminating the dormant cells in their marrow and then preventing metastasis down the road. So that's really where that is headed.
And so how far do you think you are from doing these tests in humans?
I mean, it's really difficult to state how far. I mean, what we know is that we have to go stepwise. And so the first step is, can we humanize an antibody that is going to actually work as well as the antibody that we were using already? If it does, is it going to be able to prevent metastasis in mice and without causing any toxicities? And if we can basically reproduce everything that we've done already and define further the context that we would want to use it in, is it ER positive, is it triple negative, is it HER2, is it all of the above, then we can now think about how we would do a trial in a person. There you need toxicity data. I know there is a company that has an integrin beta 1 antibody and that they're supposed to be doing toxicity studies any day now in glioblastoma patients. So if they can show whether or not it's toxic, that's going to allow us to really kind of push this forward faster. If we have to establish all the toxicity data on our own, I mean, obviously we need a lot of funding to do that and you know, some kind of industry backing. And this is how it can end up taking a lot longer to get it into a human. So we just have to see what happens.
So you kind of answered this, but people are wondering if there are any trials available to get into. So it's good to know that people are interested and maybe willing to participate in the future. So another question is,
there are a couple of trials that are focused on targeting dormant cells in triple negative breast cancer patients, and they're both based at Penn, and they have probably pretty restricted inclusion criteria. One of them is called the CLEVER trial. I think it's CLEVER, and it's a drug that's used in malaria treatment. That they're leveraging that to try to kill dormant cells in bone marrow and then see if that prevents metastasis. So that's one trial, and that again is at Penn, University of Pennsylvania. And then they were going to have a second trial, which the drug got pulled by the drug companies, and they're now reworking that trial into something new. And I don't know if that one's on board yet or online yet, but that's something to keep an eye on.
Great. And another question, do you anticipate integrin inhibition plus chemo to potentially be used to stabilize cancer in already metastasized cancers?
Yeah, I mean, it's definitely possible. I mean, metastases don't all show up at the same time. And if we could prevent additional metastases from showing up, maybe we can then kind of control the one that's there until, and keep somebody healthier and alive for much, much longer. So could we use this kind of approach to kill the seeds of future metastases when as soon as the patient prevents with one metastasis? That is something we don't know the answer to. It's certainly something worth testing, and I wouldn't rule out the biology of these things perhaps being similar so that we could actually apply these kind of therapies to prevent future metastases in a patient that presents with like one metastasis, for instance.
So people are curious, are you talking kind of off the top of your head, if you can guesstimate, are you talking a few years or like over 5 years or over 10 years?
I would say more than 5 and hopefully less. Yeah, more than 5 and less than 10 would really be my hope. I want to like push things. If it's only up to me and I had unlimited resources, it would go a lot faster. But the fact of the matter is it's not. So it's hard to estimate based on, I don't know how limiting resources will be and how, how a lot of these unknowns are going to go. But I don't want to promise anything without knowing those things.
So someone is asking, do we know anything about the interplay of dormant tumor cells with chemo resistance in metastatic patients?
Yeah, that's a good question. We don't really know about that in metastatic patients. I mean, one logical conclusion you could make is that when somebody presents with metastasis and then you start treating them and then other metastases emerge, what did they come from? One possibility is they came from cells that were just already growing but below some kind of limit of detection by imaging. And then, they just take a little bit longer to catch up. And in fact, their outgrowth could be triggered by the emergence of the first cell. So, I mean, one thing I would imagine is that you have dormant cells throughout, one of them wakes up and starts growing, and then the things that growing metastasis is releasing, as well as the disruption that is causing in the body, is sufficient to then block suppressive cues elsewhere and trigger other dormant cells to start emerging. And that may be why you see oftentimes one metastasis show up and then multiple metastases show up. So that's one answer I would give. But as far as the molecular mechanisms, that's still kind of being, the molecular mechanisms and how they relate to drug resistance are still being worked out.
So just one more question. So if money was not an issue, what would be your focus?
Yeah, so I mean, I've been very fortunate to have funding from the DoD that early in my career that has really allowed money not to be as big of an issue for me as it would be for a lot of people just starting out and starting their labs. And so what that's allowed us to do is to not have to pick one approach to target dormancy. So what I talked to you about today was therapeutic resistance, and how we can overcome therapeutic resistance of these cells, and how it's not tied just to their dividing. But we've also come up with approaches to figure out how these cells survive and to target that. We figured out what keeps the cells dormant and whether or not we can keep them dormant. And we've also more recently been focusing a lot on how these cells evade the immune system and how we can make the immune system actually find them or engineer immune cells that are better able to find and kill them. So by not taking just one approach, we have multiple approaches, at least 3 of which look really promising therapeutically. And so if money was not limiting, I would just like continue on those 3 paths and continue to move them forward to the point where we actually get things into the clinic, test them, show whether or not they work, and then hopefully we're right about all of the biology that we're figuring out, they work, and then we can actually have drugs that now are targeting dormancy and preventing metastasis. So we're continuing, not from a basic perspective in the lab, to not be limited, and we're hoping that that's going to continue to push us into really finding lasting solutions to this problem.
So one final question from the audience. Do you feel there's any value in examining bone marrow in DTCs in metastatic patients who achieve NED status to potentially predict further metastasis?
I think that there's value, whether or not it would predict future metastasis. To my knowledge, that study has not been done. I know that there's been a study that's like looked later and shown that is predictive. If they're still around 5 years later, for instance, that's predictive of metastasis in the future. If they're around after treatment, that's predictive of metastasis in the future. I haven't seen a study that has looked at that context. Patients who had a metastasis, stage 4, went to no evidence of disease, did marrow aspirates, and then looked for future recurrence. I mean, I think that what we know is that the vast majority of patients who have define metastases, even when they become no evidence of disease, we know the vast majority are going to recur. So I don't know about the value in predicting, but the value in isolating those cells and profiling them to learn about the disease that still is around in order to target it, I think there's a lot of value in that.
Well, thank you, Dr. Ghajar, for a very informative program. Thank you for your time. Best of luck to you in your research. Thank you all for participating and submitting questions. If you wouldn't mind taking a moment to fill out the survey at the end of the webinar. It may take a minute or so for the survey to download. We really appreciate your feedback. Dr. Ghajar, thank you so much. We really appreciate it.
All right, thank you all.
Thanks,
Thank you all for the great questions. That was a lot of fun. Thanks.
Thanks. Bye-bye.